Good evening, everyone. Today we will start with the new topic circular dichroism spectroscopy. Okay, so we'll look into these details a bit later. So let us first look into the absorption spectroscopy, what we have learned till date, because this is an extension of the absorption spectroscopy. Okay, so till date, till now we have seen in the absorption spectroscopy we have a source and then we have a monochromator. Okay, then this monochromator, it is associated with another uh, slit which allows only a specific wavelength, uh, specific wavelength of the radiation to pass through. So the first one, both of these, they form the part of the monochromator. The first prism, the prism, it decomposes the light, and this slit, it allows the selection of a specific wavelength. So the is a light of specific wavelength, it comes out over here. Then we have the sample, the light which is coming out of the sample, it interacts with the sample, which if it is liquid, they are kept in the cuvette, so interacts with the liquid sample, and then the radiations goes out, and we have a detector to capture the radiation. Okay, so light comes from here, then over here there is a breakdown of the light. Only a light of specific wavelength, it comes out of the slit, it passes through the sample, and then it reaches the detector. Okay, so this was one of the arrangement of the absorption spectrometry. There was another this thing, um, way of doing this absorption spectroscopy. That was, we had the source of light, then we have had collimator, okay, then through this collimator the radiation falls onto the sample, and then we had this monochromator, and then we had the detector, monochromator. Okay, over here, after the so we have light which goes on like this. So collimator, it condenses the signal, and only a small beam of light, it passes through the sample, it falls onto the prism. Okay, now prism, it breaks down the signal. Now from here, we have through this exit slit, we have a radiation which passes out, and it falls onto the detector, and that is detected. So circular dichroism spectroscopy, it is also a kind of absorption spectroscopy. Okay, now let's see what is it. So the circular dichroism spectroscopy, it is a absorption spectroscopy. Okay, so it is just another variant of this absorption spectroscopy, and there will be some changes in the optical parts, and then we will be having this thing. Okay. So a dichroic material, it either splits up the visible light into beams of different wavelengths or different polarizations. So dichroic material, what it is doing, it splits up the visible light into beams of different wavelengths or different polarization. So circular dichroism means uh, it will rotate the light things and it will split up the beam accordingly. So in circular dichroism, we use this property that the visible light is being split up into different polarizations. Okay. Now, what is a polarizer? If you look into any wave, so suppose I am having this bulb and radiation are being generated. Okay, and this is being generated. The radiations are being emitted in all the directions. Okay. Now, we all know that light waves, they are transverse waves. What are transverse waves?
Which travel perpendicular to their like direction of motion.
Yeah, exactly. Okay. So this magenta line, this is in perpendicular plane to this what do you call to the drawing plane where I am drawing, and this blue line, it is in plane over the board where I am drawing over the screen. Okay. So if you see, suppose if this blue line, if it is in say the electric field, then this purple color line, this is the magnetic field. So any light wave, when it is traveling, it is having electrical field and magnetic field which are perpendicular to each other. Okay. Now under this condition, so what is happening, this is only a single beam. Now suppose in a 360-degree this thing, we can represent this source. This is just a schematic representation for your understanding. Okay. So suppose there might be an electrical field which is moving up and down. There may be another this thing which is perpendicular electrical field which is moving perpendicular to the first one. Then there might be another one in this direction. Okay. There might be another one in this direction, and all of this propagation, whatever it is having, whatever we are getting, these are, see, in all the directions. Okay, so I have drawn in a 2D plane. If you consider it is as a 3D, it will be having these projections in all the directions in a spherical way. Okay, now when you pass this radiation through a plane polarizer, then what happens? Only field in one direction, in this case, say those who are which are those waves which are having uh, electric field, parallel to this or over this drawing sheet, they only passes out and they move forward. Okay, and the all other waves or all other light waves which are moving, they are what you call rejected by this plane polarizer, and the light which we get across the plane polarizer, it is regarded as plane polarized light. Okay, plane polarized light. Okay, so it is basically a linear polarized light. Okay, it is basically a linearly polarized light. Okay, now if you look, it is having an electrical field and magnetic field, and then we get this wave pattern. Okay, so this is this thing, and here we are not talking about the magnetic field as such. The reason being, usually the electrical field of a wave is quite high as compared to the magnetic field, so we can neglect that magnetic field. Okay, so the light waves which are moving in only one plane, it is regarded as plane polarized light or linearly polarized light. Okay, there is another kind of polarization which is regarded as circularly polarized light, which is regarded as circularly polarized light. Okay, so herein, if you see, the continuously the direction of the light, it is changing, and it is changing in a helical way. So if you look into the direction of the propagation k, it is moving forward, but it is moving in a circular way. Okay, so so if you look from this end or from this end, from the direction of the propagation or against the direction of the propagation, then what you will see from top, that you are having a circle, and then you have electrical polarization in all the, so it looks like a circle and it is having structure like this. So it is because from this direction of the propagation we are getting different movement as the light waves, they are moving forward, they are creating this helical like structure. Okay, otherwise they are moving in a circular motion across this direction of the propagation. Okay, so hence they are regarded as circularly polarized light. Okay, so circular dichroism is the dichroism that involve circularly polarized light. Okay, and circular dichroism, it is in short regarded as CD spectroscopy. Circular dichroism spectroscopy, it is regarded as CD spectroscopy. Okay, and it deals with the differential absorption of left and right handed circularly polarized lights. Okay, now why this is important? This is important because the analysis of the circular dichroism spectra, it allows you to understand or to gather the structural information of the proteins in biology. Okay, so basically it helps us in the secondary structure determination and protein folding and misfolding. Okay, now the circularly polarized light, they are categorized into two categories: left-handed circularly polarized light and right-handed circularly polarized light. So if you see from the top or from the direction of the propagation, so you will find the light is moving as in the anti-clockwise direction. Okay, whereas if you look into the right-handed circular light, you will see that the electrical field, it is moving in the clockwise direction. Okay, and if you see, it is moving in the anti-clockwise direction, and then it is advancing, continuously advancing, slowly advancing, and then it forms a helical structure in this way. Whereas in the right-handed circular light, the helical structure like thing, or the path of the light, it is in this fashion. Okay, so in the left-handed one, you will start from the base and move towards the left and slowly go up. Okay, whereas in the case of right-handed circular light, you will start with the right hand movement and then it goes like this. Okay, so this is the another representation of the electrical field vector, how it is they are moving. Okay, and they are regarded as right-handed circularly polarized light, or CPL. Okay, similarly for the left-handed circularly polarized light, it is regarded as LCPL. Okay, now why it is important? This is important because if there are any chemical compounds which are optically active, you can analyze those molecules. Okay, so one of the primary requirement is that the molecules should be asymmetrical, or they have chiral center, or they are the enantiomers. Okay, so a chiral molecule, we all know it has a non-superimposable mirror image. Okay. Now, if you look into the UV spectra of the camphor enantiomers, both of them, they are having same molecular chemical formula. Okay. And if you see the spectra, both of them, they are giving same spectra. However, in this circular dichroism spectroscopy, if you see, there is a change in the nature of the uh, what do you call nature of the spectra. Now, if you look over here, this is plus camphor means dextro-rotatory. This camphor is dextro-rotatory. That means, suppose we have a light source, then we allow it to pass through a polarizer, plane polarizer. So we get a light over here, and then we pass this through the sample. So what it will do, suppose the light over here, it is in this direction. Okay, so what the dextro-rotatory samples will do is that it will move the electrical current in the clockwise direction, and this clockwise direction movement, it is called dextro-rotatory. If it is on the left hand side, then it is called levo-rotatory, and it is given by this minus sign. Okay, now uh, let's look into this the mechanism or the instrumentation of the uh, this CD spectroscopy. Okay, now we have the source as usual, so you can use whatever the source you have used in what you call in your UV visible spectroscopy in the visible range. No, you can use anything. Then you have a monochromator. So you have a broad, what do you call, white light. It falls onto this monochromator, and only a specific wavelength light it passes through. Then we have a photoelastic modulator. We have a photoelastic modulator. And there are two types of photoelectric modulator. One generates or converts this monochromatic light into LCPL. This is this one. So this is LCPL and this is RCPL. Right-handed circularly polarized light, left-handed circularly polarized light. Okay. So there is an electronic this thing electronic circuitry which changes the position of the photoelastic modulators such that at one instance you get left circularly polarized light, and the other instance you get the right circularly polarized light. Then what happens is that these lights, circularly polarized light, so if you see alternating L and R, so this circularly polarized light, they are allowed to move through the sample. And while they are moved, what happens? The left-handed circular polarized light and the right-handed circular polarized light, they are differentially absorbed. Okay, they are differentially absorbed. Okay, what does that what does that mean? That means that it is quite possible that the left-handed circularly polarized light will be absorbed more by the optically active sample as compared to the right-handed circularly polarized light, or vice versa. So if you see, in this case, it has been shown that the amplitude of the left-handed circular polarized light, it has decreased, whereas there has not been a substantial change in the intensity of the right-handed circularly polarized light. Now there is a PMT. Okay, why we use PMT? Even though PMT does not have wavelength information, this is because over here we are choosing a we are using a monochromator, so we are basically using a specific wavelength light. So when you are using specific wavelength light, then there is no need for the information of the wavelength. Okay, so this is one of the important thing. In normal UV visible spectroscopy, you can do the analysis either at a specific wavelength, which we call lambda max, or we can go for wavelength scanning, where information about all the wavelengths are required. So in that case, the detector arrangement is totally different. Suppose we were just interested in the lambda max, which we discussed about in this thing, in the colorimeter. Okay, so over that we are just using a specific wavelength light. So in that case, you can just use a PMT. There is no need for you to go for the other costly detectors. Okay. In addition, what happens? You have this PMT which will amplify the signal from here. Okay. So your electronic circuitry, it will have the information when this photoelastic modulator, it is generating the left circularly polarized light. So it will also control the PMT, and whatever the signal will be uh, recorded by the PMT, it will be stored as the absorption for the LCPL, and then when this PM will be generating RCPL, it will be recording it as the RCPL. Okay. So in this way, it will be having two kinds of absorptions. One is AL and the other is AR. Okay. AL is the absorption due to the left circularly polarized light, and AR is the absorption due to the right circularly polarized light. Okay. Now, as we are telling that differentially absorbed circularly polarized light. So basically what we want to see, we want to find out AL minus AR or vice versa, depending upon whatever you are giving, right? So AL minus AR, it is needed. So this is calculated basically in this way. Okay. Another important thing is that when you are using this photoelastic modulator, just before this there is a linear polarizer, and this linear polarizer, they are usually made up of nickel prism or Glan Thomson prism or tourmaline crystals. Okay, among these, Glan Thomson prism, it is most commonly used. Okay, so this polarizer, it is basically integrated with this polarizers. Okay, so no, with this photoelastic modulator, the polarizers, they are integrated with the photoelastic modulator. Okay, so hence in this diagram it has not been shown. Anything you would like to ask? Do you guys have any doubts?
No, sir.
Okay, okay. Now the speeds between the right and the left polarizations differ when they pass through the samples. Okay, then the next is the wavelength of lambda L and lambda R, they are not equal, and the extent of absorption, uh, which is regarded as the molar extinction coefficient, epsilon L and epsilon R, they are not equal of the polarized lights. Okay, they also differ. Then circular dichroism spectroscopy, it calculates the difference in the molar absorption coefficient. Okay, so if you remember carefully, so during our UV visible spectrophotometry, what we said, we had A equals to epsilon BC. Okay, so A was the absorption. Now we have talked that there is a change in the absorption. Now why the change happens? The change happens due to the molar absorption coefficient. So basically, because if you look into these other two components, B was the path length and C was the concentration of the solution, they will remain the same for both LCPL and RCPL, right? So what is basically changing? This is changing. So basically, while you are measuring the absorption, change in the absorption, so what it is happening is that you are basically measuring the difference in the molar absorption coefficient, and this factor delta epsilon, it is regarded as the molar circular dichroism. Molar circular dichroism. Okay, the CD spectrum is often reported in degrees of ellipticity theta, which is a measure of the ellipticity of the polarization. Okay, now what it is saying, it is saying, suppose here this red one and this blue one, these are the primary wavelengths. Okay, primary circular lights. So it is having an electric field over here, and since they are concentric, okay, and the other one it will be having whatever you say, okay. Now what you are trying to do, you are trying to find out, say, ER minus EL, okay. Say for example, at these locations there is a constructive electrical interaction. So due to that, what happens, this purple color line, you are having an addition component, and in this perpendicular direction there is a um, what do you call, there is a subtraction of the electrical fields, okay. So what you have now, you get a theta, right? You get a theta. So what you are getting, ER minus EL, and here at this point you are getting ER plus EL. So it is basically forming an ellipse, okay. It is basically forming an ellipse, this ellipse, right? Now this information, this theta, it is dependent on this difference. If difference is quite large, that means in other words EL minus EL is small, then this length will be small, so your theta will be smaller, okay. And if this difference is high, the value is high, then it will move there and there will be an increase in the theta value, right? So that is what it is telling. The CD spectrum is often reported in degrees of ellipticity theta, which is a measure of the ellipticity of polarizations, okay, ellipticity of polarizations. So it is what it is given, elliptically polarized light, purple, is the superimposition of LCP, left circularly polarized light, red, and RCP, right circularly polarized light. Theta is the angle between the magnitude of the electric field vector at its maximum and its minimum, okay, at its maximum and its minimum, okay. The change in the polarization is usually small and the signal is often measured in radians, where theta is equal to 2.303 by 4 AL minus AR, and it is a function of the wavelength. That's why we have seen that we use monochromatic light. Okay. Theta can be converted to degrees by multiplying by 180 by pi, which gives theta is equal to 3.298 multiplied by delta A. Okay. So if you see, uh, they are all being converted, and the constant which comes out to be it is 32.98. Okay. The historical reported unit of CD experiment is molar ellipticity, okay, and it is given by this, see, square bracket theta, which removes the dependence on concentration and path length, okay, and it is given by 3298 multiplied by molar difference in the molar extinction coefficient, okay. The constant 3298, it converts from the unit of molar absorptivity to the historical units of degrees multiplied by centimeter square per decimal dissimilarity. Okay. Now, when you do this spectroscopy, CD spectroscopy for a protein, you get this kind of curves. Okay. Now, this red curve, it is for the alpha helix, and you will find that there are three peaks, one at no, one positive peak at 190, and then we have 208 nanometer, this one, 190 is this one, and 222 nanometer. So one positive peak and two negative peak at these locations. These locations are important. This will not change. Okay. In beta sheet, we have a peak at 196, which is a positive one, and then we have another peak at 218 nanometer. Okay, which is a negative peak. Then we have this random coil. In the random coil, we have a negative peak at 195, and we have a positive peak at around 212. Okay, so these are for the negative one. Now, accordingly, say we are analyzing myoglobin and we want to find out uh, what happens. In factuality, myoglobin, it is an all alpha type protein, alpha helix type protein. Okay. Now, if you look into this hemoglobin, myoglobin, this spectra, it totally matches with this alpha helix. So, at these positions, we will be getting these peaks at these said locations. We will get these peaks, and by just analyzing the nature of this curve, we can say that yeah, this myoglobin, it is having an all alpha helix structure. Okay. If it is mixed alpha plus beta, it will be a combination of alpha and beta curve. And if you see this lysozyme, yeah, this red one, so you are having this, then you have a peak over here, then you have another peak, and then you are having a, so if you see, it is in between your alpha helix and beta helix. Okay. Then we have this triose phosphate isomerase, mostly alpha and some beta. So this is the green one. So if you see, it is having this thing, and then it is showing near alpha this property. So this is your mostly alpha, and it is having some beta. And then if you look at the intensity level is quite different, and that intensity is because your beta sheets, they are pulling down the what do you call, they are pulling down the intensity levels, and hence from there you can say that it is mostly alpha and there are some beta. Okay. Alpha sheet, if you see the alpha sheet, it will be very high. Okay. Now if it is mixed alpha and beta, the beta component and this alpha component, they are almost 50 and 50. So the pull down will be marked. So this red line, if you see, the intensity is quite low, right? And lastly, we have chymotrypsin. In chymotrypsin, we are having all beta. So you see this one, sorry, this one, you can see you are having this peak. Okay. So in this way, you can tell whether a protein structure, it is having an alpha helix structure, or it is having a mix of alpha helix and beta sheet structure, or it is having what type of mixture, whether the alpha component is higher in that mixture or beta component is higher. You can look into that, or if there is a more of beta component, all of beta component, and like this. Okay. So in this way, you will be able to find out what are the different what are the different types of yeah, protein structure which are available within the stuff, okay, within the protein mixture, protein solution mixture, okay. So with this we completed this CD spectroscopy. If you have any questions, please feel free. No?